Hello, everyone. Good morning, good afternoon, good evening, wherever you are. Welcome to the 9th week in the long-term animal research seminar series. We continue this week with Dr. Alastair Wilson. And before I introduce Alastair, I will make a couple of announcements. First, if you're participating via Zoom, you should see a Q&A tab on the top or the bottom of your screen. If you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. And at the end of the talk, we'll go through those questions, starting with the ones with the most votes. However, if you have a clarifying question that you feel like needs to be addressed during the talk in order for you to understand something, you can type clarification in capital letters at the start of your question. And Alastair has said that he'll do his best to answer those questions in real time. Second, recordings of all the talks will be available on YouTube shortly after they conclude. So if you need to leave early or know others who are unable to attend live, the talk will be available for viewing and reviewing after it's complete. Now I'm very pleased to introduce today's speaker, Dr. Alastair Wilson, Professor of Evolutionary Biology at the University of Exeter. Alastair received his bachelor's degree from Cambridge before completing a master's degree at King's College and then a PhD at the University of Guelph. He then held a series of postdoctoral and fellowship positions at the University of Edinburgh and the University of Exeter, where he is currently a professor. He has received numerous awards, including the 2020 Mary Lyon Medal, which is an award from the Genetics Society that recognizes outstanding genetics research from scientists who are in the middle of their careers. And Alastair, who leads this really exciting research program that uses both wild studies of sheep and badgers, as well as lab studies of fish to understand how genes and environment shape phenotypes and how natural selection leads to evolution and what mechanisms might prevent it from doing so when we think it otherwise would. And something that I really admire about Alastair's work is his ability to communicate quantitative genetics theory and applications to audiences like the one that this series caters to, whose primary focus may be behavioral ecology or endocrinology or life history or evolution at the phenotypic level, and many of whom study wild systems. And if you ever felt like you wanted to have a better understanding of how quantitative genetics might be relevant to and applied in your study system, but have also found that literature to be somewhat unapproachable, and I recommend 2 papers of Alastair's that really helped me feel more confident in approaching these concepts. One is a 2010 paper in the Journal of Animal Ecology titled An Ecologist's Guide to the Animal Model, and another is a 2009 article in Trends in Ecology and Evolution titled What is Individual Quality and Evolutionary Perspective? Anyway, like I said, those both really helped me feel more confident in the area, and we'll learn a lot more in this next hour too. So sorry for going on a bit long there. I'm now excited to welcome Alastair and turn things over to him.
Okay, thank you very much, Matthew, for the invitation. I'll just find the screen sharing tab and see if I can find my talk. Here we go. Okay, so thank you very much for the introduction. That's very kind. I now feel that I'm nicely set up to fail, as I do a terrible job of trying to explain quantitative genetics to people. But I guess I'll see what I can do, and I'll try to shed more light than confusion, but I make no promises, I guess. So, title of my talk then is Evolution in Wild Populations, from Genes and Environments to Genes as Environments. And hopefully the kind of rationale for that subtitle there will become clear as we progress. But I guess I should start at the very beginning by saying that what I'm really interested in is how traits evolve. And if I'm honest, I'm not really interested in, or so interested in, neutral evolution. I'm really interested in how traits evolve under selection, albeit not always natural selection. Sometimes I'm thinking about artificial selection. And I do this, or in my group, we do this usually using the framework of evolutionary quantitative genetics. And as a starting point for that, we often find ourselves trying to answer the apparently simple question of how much variation in a population for some trait, it could be height or horn size in this population of sheep here, but how much of the variation among individuals do we think is caused by genetic differences among those individuals? And the reason that's useful and potentially interesting is that if we can do that, we can put a number on that, and we can also characterize selection on that trait. And selection is really then arising from the interaction of the environment with phenotypic variation. If we can put a number on selection as well, we can use quantitative genetics to kind of combine those two things, the genetic variation and the selection, to make some statements about how the trait should evolve, or potentially we can think retrospectively also about how the trait has evolved in the past. And that's kind of the basic idea. So what I thought I would do in this talk is I've kind of broken it into three chunks, which will probably bleed into each other a bit. But this is the outline. I'm going to spend a few more minutes talking about quantitative genetics in a very kind of general, basic way, just to try and make sure everyone's on the same page. And in that, and within the theme of this seminar series that you guys have been running, I want to kind of highlight why we're trying to use it in wild populations and the role that long-term studies have been playing in that. So that's the first bit. And then what I thought I'd do is I'd talk a little bit about a specific long-term study. This is the European badger, and I'll get onto those guys in a little bit. And that's really, in some ways, at the more basic end of what you might want to do with quantitative genetics, but it's interesting, I think, in an ecological and also somewhat interesting in an applied sense. What we're trying to do there really is work out if we can separate the extent to which genes and environments influence particular traits of interest. And then having done that, I'm going to spend the second half of the talk really, so this will be the major bulk, kind of talking about not so much a system, but an idea. And the idea really is that separating variation into genetic and environmental sources in wild populations is itself a bit problematic. And it's problematic for a number of reasons, but the one I want to focus on is that actually an awful lot of what we think of as environmental effects are arising through interactions with conspecifics. And those conspecifics, of course, are carrying genes. And so there end up being all these interesting pathways where genes carried by other individuals might be affecting the phenotypes of focal animals that we're looking at. And that's something called indirect genetic effects. So I'm going to talk about them, and I'm going to talk about them particularly in the context of animal contests and competition. I've kind of cherry-picked some bits of research from long-term studies to fit with the theme. I rather suspect I've overloaded it a bit. So just at the end of that section, I was going to talk a little bit about whether indirect genetic effects might also be important in cooperation. Depending on how the timing goes and how bored Matthew looks at that point, I might just skip over the last few slides, or I might keep going. We'll see how we go. But of course, as and when my droning on gets too much for you, you can just leave and I can't see that you've done it. So that's okay for you. I'll just be demoralized at the end when there's no one left. Okay, so quantitative genetics in the very broadest sense, what is it? Well, it's just about studying the genetics and inheritance of complex traits. Complex traits being aspects of phenotype that we think are almost certainly influenced by environments to some extent, but also have some kind of genetic basis of determination. And they're complex usually in the sense that they're polygenic. So these are not simple Mendelian traits where we can map one trait or one gene to one trait. They're things that we think probably are dependent on a whole bunch of different genes acting in different ways and distributed throughout the genome. Now, I think of it sort of as a meeting place. This is probably a bit overly romantic, but I like this thought, so let's hold on to it. I think of it as a meeting place between geneticists and ecologists. And I have to admit from the off, I'm not really a geneticist and I'm not really an ecologist. I think my role in this has become that of a translator, where I'm able to talk to people in both camps. But I said we start often by thinking about how much trait variation in some population might be caused by genes. And from there, we can go in two directions. So we can talk to the real geneticists who actually want to work out, well, where's this signal coming from, right? So you've shown me there's some variation that's down to genes, but now what are the genes? Where are the genes? How many are there? Where are there in the genome? And so we need to get into the genomics, the genome structure. We need to think about maybe mapping the genes and maybe doing some functional genetics to do that. So that's one way we can go. We do a little bit of that in my lab, but to be honest, I never quite embraced 21st century genetics. I mean, I'm still working on it and I hope to one day, but I'm still kind of living in the past. And I find it easier to think in the other direction. So the premise, or arguably, I think you could say the conceit of classical quantitative genetics is that if you're really interested in trait dynamics, right, rather than genetic architectures per se, you might be able to ignore all that detail, and you can just take sort of some kind of statistical description of the inheritance of traits or phenotypes. And you can work upwards. You can use that description to think about, for example, whether selection on the trait is going to cause a response. How fast will that response be? And then we can talk to, population ecologists, for instance, and think about what that might mean for these populations. So I don't know, let's take a trivial example, well, not a trivial example, but one that's kind of easy to understand. We could think about whether a population threatened by a changing environment might actually be able to evolve traits in such a way that it can adapt to persist in that changing environment, or maybe it's going to get wiped out because it can't evolve to keep up. So we can start to think about the population level consequences and really work with ecologists in that way. So that's more the kind of looking upwards side of things that I do, and that's really what evolutionary quantitative genetics is about to me. Now it looks like this. So to me, this is, of course, a thing of beauty, and I can just look at that and know exactly what it means, obviously, because I was born reading Greek and doing matrix algebra. So if you're the kind of person who isn't super keen on maths, then you might look at this and hopefully at this point you're thinking, wow, that guy must be really clever to handle all that complicated stuff. So just hold on to that thought, and I suspect you'll be changing your mind within 5 or 10 minutes. But hold on to it for as long as you can. If, on the other hand, you're good at maths and or you've had training and exposure to quantitative genetic theory in the past, then you'll probably recognise most of these equations. And I guess I can shortcut the whole talk by saying that this is all the real science underpinning what we've done. So you can now have a snooze for the rest of it and just look at the pretty pictures. But go back to the people in the first camp again. This looks complicated, and it is complicated, but the dirty little secret about quantitative genetics is that what we love to do is take simple ideas and make them as complicated as we possibly can. That's just because we love the complication, we love the complexity in a way actually that arguably I would say behavioral ecologists love simplicity. So we tend to make things quite complicated. But here's what you really need to know, right? All of these equations are they're just different representations with different biological kind of nuances. But the premise is simple. It's that we can predict adaptive evolution, so that's the response to selection, as the product of two things. Something that captures the core principles, if you like, of inheritance of the phenotype, so that's the genetics, and then also something that captures the selection acting. I'm going to talk mostly about the genetics today. I'll mention selection a few times, but I'm not really going to be presenting any detailed selection analyses. Okay, so here's our simplest model, and this is probably familiar to most people. It's called the Breeder's equation, and it's usually attributed to an American geneticist called J. Lush, I think, dating to the 30s, so it's been around for a long time. R here is a response to selection, so that's how much does the mean phenotype change after a single generation of selection, and that's predicted as the product of H squared, the narrow sense heritability, I'll come back to that in a sec, and S, which is a selection differential. So S is just a measure of the strength of selection, or really the strength of directional selection acting on a trait, and in wild populations we might get it measures of selection essentially in a correlative way. Of course, selection needs to be causal, but being good biologists, we ignore the rule that causation doesn't equal correlation whenever it's convenient for us to do so. We look at the relationship between fitness and a trait value, and in this case you can see there's a nice linear relationship, and so we'd say this trait, whatever it might be, is under positive directional selection, and the covariance between those, or we could use the slope of the regression line, gives us a measure of selection. Okay, so we can go out and do that in wild populations, provided we can measure traits and provided we can have some proxy of individual fitness. What about heritability? So heritability is really it is a standardized measure of genetic variance. And what I mean by genetic variance, I should kind of be very clear about this for people who aren't used to quantitative genetics. In quantitative genetic terms, genetic variance is phenotypic variance that we think is explained by genes. All right, so I'm not talking about molecular genetic variance here. I'm talking about how much of the variation do we think is down to genes. And heritability is just the proportion of that. So it's it's the amount of genetic variance, or strictly speaking, additive genetic variance, divided by the total phenotypic variance that you see in the trait. So it's going to be a number between 0 and 1. And in terms of how you estimate it, there's lots of complicated statistical techniques, but bottom line is that they all rely on a really simple premise. And the simple premise is this: if individuals that are closely related, and so by definition tend to share more of their genome, also are systematically more similar to each other at the phenotypic trait you're interested in than if you were to say pick 2 random unrelated individuals from the population, then from that we can infer it's very likely that genes play a big role in shaping the phenotype, right? So individuals that share genes share phenotypes. And all we're doing really in kind of pedigree-based quantitative genetics is estimating the strength of that relationship, and that can be used to infer the amount of genetic variance for a trait or for a multivariate phenotype. The catch, of course, is that you need to know how individuals are related to each other. And that means that historically, empirical quantitative genetics, so I should say the kind of theoretical models that I've shown you have been, they really are, I suppose, our fundamental evolutionary models for how phenotypes evolve. But how do we parameterize them? Well, to do that, we need to be able to estimate the genetic parameters, things like heritability, but also genetic correlations when we think about 2 traits, or G matrices if you go fully multivariate. And to do that, as I said, we need to know pedigree information or relationships. So for a long, long time, empirical quantitative genetics was really, I'm going to say stuck, which sounds pejorative, which I don't mean, but was limited in the sense that it was something you could apply to lab studies. So you could work on model organisms in mice or Drosophila or Arabidopsis, for example, but also clearly important in the livestock industry where you're controlling the breeding decisions. And so almost by definition, you know who's related to each other. But it's got a much more recent history in terms of application to wild populations in situ. And of course, there are studies that happened before 20 years ago, but to a first approximation, there's been a massive growth in this field in the last 20 years. I think there's three drivers of that. The first is starting in the mid-90s, things like microsatellites. So we had molecular tools that meant we could start exploring relationships among individuals in a population from a pretty low base knowledge. And of course, now we've got the opportunity to do even better with high density SNP genotyping and things like that. So that's one thing. The second thing has been development of statistical models that actually basically are better able to handle complex, messy, unbalanced pedigrees and data sets that are typically what we get in wild populations. And in particular, I guess, all the examples that I'll show you today have used something called the animal model, which is just a form of mixed model that we can use to estimate genetic variance and related parameters. But the third factor, certainly and conveniently for this seminar series, has been the role of long-term individual-based studies. And so long-term studies, of course, come in lots of different flavors, but for current purposes, the ones that have mattered are the ones where individual animals have been tagged, marked, and tracked throughout their lifetime. So we usually know when they were born or hatched, when they died, and hopefully we have lots of phenotypic data on what's going on in between. And I suppose there's a number of factors that have made these so important. So I'll rattle around them in kind of clockwise order from the red one at the top there. The first and foremost is quantitative geneticists follow the data, right? Our methods are data hungry. So we need high volume data on phenotypes. And it's a lot easier, let's be honest, it's a lot easier to wander up to someone who's been collecting data for 20 years and say, can I play with your data, than it is to wait 20 years to use your own. So there's no escaping that's kind of important. The other thing that's happened in long-term studies is that pedigree has often been available. So if you think of bird ringing studies, sure, there's some extra pair paternity, but actually you are ringing birds at the nest. And if you assume that the extra pair paternity is not too high in the first instance, then actually you're building up social pedigrees as you go. Same thing happens a lot with mammals. You can often infer the mum just from your observational work in the field that you're already doing. And then from the 90s onwards, people, of course, have been able to check paternities and also explore pedigrees and relatedness in situations where you can't maybe infer the mum. Lifetime fitness measures. So I mentioned going back a little bit that we want to ideally combine our measures of genetic variance with selection. Well, where do you get the best proxies of individual fitness? You get them from long-term studies where individuals have been tracked from birth to death. And so we know, for instance, the number of offspring they produce, which gives us a fairly robust, of course it can always be criticized, but a fairly robust measure of fitness relative to some other things that we might use that are defined over a shorter term. The next two, environmental change and rich biological knowledge. I mean, that kind of goes without saying that in long-term data series, there's often interesting things going on in the environment. Sometimes that's been the rationale for setting up the study. And there's usually a whole host of people who've worked on those projects who are very keen to share their knowledge with you. So that matters because ultimately quantitative genetics is a tool here. So we need to develop interesting biological hypotheses and questions. And that's much easier to do when you've got interesting environments happening and interesting people to talk to. And that leads to the last one, which I think is, it's never really said in the papers, you know, when we talk about this, but actually in my experience, the thing that's been most important has been the incredible support and interest and open-mindedness, actually, of ecological PIs who've led some of these projects. They're interested in people coming in and applying, you know, they might be behavioral ecologists or life history people, but they're actually interested in people coming in and applying different approaches and complementary perspectives to perhaps address the same sort of evolutionary puzzles. And so that's been super important, I think, that it's perceived as a collaboration and not people coming in and just pirating data. Certainly that's the way my career has developed. So I started off in this line of thinking, at least. I was going to be a molecular ecologist. I was like, oh, I'm going to do loads of PCRs and run gels and do all kinds of stuff. And then after 3 years of my PhD, standing in a lab wearing wearing a lab coat and wielding a pipette, I just thought, never again. And so I started using the data I've been collected to see if I could, instead of looking at population structures, think about relationships among individuals within populations. I was working on salmonid fish at the time, and then starting to think about testing some quantitative genetic ideas about life history traits and growth in some of these salmonids. And then I moved to Edinburgh because just at the time I was messing around with what were quite small fish data sets that I'd collected myself. I noticed that other people in other places seemed to have access to information on thousands and thousands of animals, and someone had built pedigrees for them. And I thought this would be an easier way to do it. So I was lucky enough to move to Edinburgh, where I ended up working on the Surrey Sheep project and also a little bit on the red deer of Rum. And at the same time, actually also doing some work on a bighorn sheep project run by Marco Festabianchit out of Sherbrooke in Quebec, which was interesting as well. And then it kind of went from there, because the thing about quantitative genetics, once you start it, is once you've figured out the kind of slightly complicated statistics, you realize it's entirely general. So the theory applies to any trait in any sexual diploid. And that means that you can explore questions in a very general way, and or you can explore lots of different systems. And so one of the really nice things I think that this field has enabled me to do is to work with ecologists from all over the world on all kinds of data systems. And at some point, of course, there's things you want to test, with or ideas you want to test or methods. So you start making up data, but we don't call it that. We call it simulation. So I am very proud to have published a paper on unicorns. And in fact, I've also published one on the wild haggis of Scotland, which is depicted in the bottom right there. And if you don't know what a haggis is, then that's okay. I've eaten quite a few of them, and I still don't really know what they are. But you can Google and they'll tell you. The point of this slide, though, is to say that while these long-term studies are amazing and they are incredible, they also have limitations. So I'm not going to dwell on them today, because it seems like we should be celebrating the long-term projects. But a big part of what we do now actually is we get wild animals, usually small ones because they're more tractable, and we move them back into the lab. And that's because there are some things where we really do want to study, if you like, the genetic variation that's we hope is characteristic of wild populations, but we do need to get perhaps more balanced data sets or more complete data sets. And in particular, the last few years have been working a lot on behavioral traits, and getting high volume behavioral data in the field, as many of you will know, is really quite challenging. So here's where we are now. About half my group work on guppies, actually, that are descended from animals we collected in Trinidad. But we're working on a host of other organisms, including the badgers, which I'll talk about in a second. And then just in the bottom right there to flag that, although this isn't, I'm sure, what you had in mind, not all long-term studies are actually of wild populations. And if you are interested in what's happening in long-term populations generally, you could do a lot worse than think about what people have done in the livestock industry, for example. So anyone who believes life history trade-offs pose inescapable constraints should go and talk to a pig breeder for 5 minutes, and you'll realize that selecting to increase the number of offspring, the size of offspring, and everything else simultaneously is not that hard to do. So, you know, there are some things in other fields that I think we can learn from. And we should do. So I have one student working on racehorses, and if that pans out, then I'm totally retiring in a couple of years. But at the moment, we can't predict the winners. So we'll see how we go with that. Okay, so that's enough preamble. We should talk about some actual science. So I'm going to talk about these guys. These are the European badger, Meles. Yeah, and that little inset is a picture of the nasty thing that lives in it, which is a mycobacterium. So it's bovine tuberculosis bacteria that lives in it. So I realized I didn't really know anything about North American badgers. And so I suspected it was possible you guys might not know anything about European badgers. I found this tweet, which I quite liked. But I suppose ultimately it just really plays to stereotypes of English or British people living either in castles or thatched cottages, neither of which I'm privileged enough to live in. I just have an ordinary house. So there we are. But I guess there is something undeniable in the fact that our badgers look nicer than yours. And that's just, you know, where it is. But as biologists, we also know that they're mustelids. So bottom line, if you get up close to a European badger, you're going to find it's pretty stinky and pretty bitey, like all other mustelids. And there's not much getting away from that. So it's medium-sized as this group go. They're actually quite variable in size, and they vary a lot during the year as they kind of put on weight and lose it. But adults can be anywhere between 6-7 kilos up to about 16-17 for a big male at his fattest, I guess. They're crepuscular, so they're active at dawn and dusk. Omnivorous, clearly they're in the carnivora, and they will take small, you know, rodents or frogs or things if they can catch them. They'll also eat berries. Some estimates, though, suggest that about 80% of their diet, at least in England, is earthworms. So that's the thing they really love, and that means that you find them at high density in kind of soft, rich soils that they can dig in. Exactly the kind of places where you'd find, for example, lots of cattle farming. You can see from the little map here that they live high density in the southwest of England, also parts of Ireland, and they really don't like Scotland. It's not that they don't like Scotland, of course, it's that they don't like highland areas with thin soils and lots of rock, because they like to dig. They're described as facultatively social at high density. Now that's a kind of vague term that could mean almost anything. If you're a social evolution type person, then what this means, I think, is that really there's no good evidence that they engage in cooperative behaviors as you might normally think of them. So there's not really any good evidence for cooperative brood care, for example. There's not really any evidence for cooperative territorial defense. They are territorial. Groups have territories, and there's lots of skirmishes, but they seem to be one-on-one type things. So what they do is they tolerate each other and or enjoy each other's company much more than most mustelids. So they'll live in groups that could be 3 to 4, up to 20, 25, 26. And there'll be multiple families in there. There'll be multiple breeders in there. There is some evidence that of suppression of reproduction that happens in those groups. But they're certainly not, you know, social in the way that some people would interpret that term. Why they're important, aside from being cute and interesting generally, they're a really important wildlife reservoir for bovine TB. And infected badgers, importantly, it can kill them. It doesn't, you know, they don't do particularly well when they're infected, but it doesn't necessarily kill them. So infected badgers can have a sort of low-level infection that goes on for years and years and years. They might not do as well as other individuals, but they can certainly survive and they can certainly reproduce. And that means that there's this ongoing potential for transmission between badgers and notably cattle. Particularly, as I said, they're occupying the same areas as well. And bovine TB is a massive problem in the UK. It's a big problem for the livestock sector. Costs a lot of money, impacts the export status of meat and dairy products, and results in animals, herds having to be culled in order to get it out of the herd and things like that. So there's a lot of politics around this, and current government strategy has been sort of largely led by targeted badger culls, and they've been hugely controversial. For various reasons. I think the first is maybe a cultural one. The badgers just really loom large in the British public's imagination, I guess, because of Wind in the Willows and things. They're actually our largest terrestrial carnivore, pathetic as that may sound to you guys. But they're sort of, they're wise and they're friendly and they're guardians of the woodland or something. So they're somehow symbolic. So people don't like that they're being managed in this way. There's also really serious welfare concerns about the way they're being shot or poisoned or whatever, and what that means for the suffering. But also in a scientific sense, there's some pretty good evidence that the culls are likely to be ineffective, and in part because culling some members of the group might increase the dispersal of other members of the group. And I think we're all aware right now of what dispersal can mean to disease dynamics. Okay, so I'm going to talk about data from this particular study at Woodchester Park, which is on the little map there is marked as the little sort of pink and black dot, which hopefully you can see. It's in not too far away from me. Actually, I'm to the southwest of that, almost at the tip of Britain, with not much between me and New York. But Woodchester then is in this high density area, or high badger density area in the southwest. And you can see it's kind of rural parkland with cattle. The big mansion there, it's an old country estate. The mansion's derelict and coincidentally houses a population of horseshoe bats that are the subject of another long-term study that I know nothing about. But I just thought you get a two-for here, so there you go. At any one time, there's 200 to 300 badgers in the study area, in 21 to 25 social groups. The social groups here are centered on what are called sets. So sets are systems of burrows that are used by the badger groups. And those sets are used for years and years and decades. So the social groups in practice are defined based on where the sets are. But at certain times, some sets might fall into disuse, for example. So the groups kind of disappear. But the core set of about 20 groups has been very stably studied for quite a long time. The basic methodology is trapping. So what happens is traps are set for 2 nights at each group location or each set, and that's done 4 times a year. So you're not catching all the badgers, but you're catching a decent chunk of the badgers that are in there. They basically are unable to resist peanuts, so you load it with peanuts, and that's how you catch the badgers. And then it's all the usual weights and measures you would take, size, condition, tooth wear is looked at, and lots of biological samples, hair with skin cells on, which is used for DNA, feces, urine, blood, sputum. If there are any abscesses on the body, which can appear as a sort of symptom of TB infection, then those are swabbed as well. I guess that's the main stuff. This is mostly done under anaesthetic, so handling a live badger without sedating it would be foolish, I guess. What then happens in terms of the TB status? It's a little bit fuzzy. So again, we all know a lot more about testing for disease than we did 6 months ago, I guess. There's false positives, there's false negatives, there's different tests. But I'm going to try and keep it as simple as possible. What's happened at the moment, or for today's purposes, they've at each capture, based on the samples taken, the animal is given a score from 0 to 3. 0 means there's no evidence of infection. 1 means that it's testing positive in a serological test, which is an antibody test. It's testing positive for TB. So that means either it has been exposed, well, could mean a false positive, but leaving that aside, it means either it's got TB, possibly just in the early stages, but potentially it's had TB and cleared it, right? So that could have happened. 2 means that actually they've actually cultured Mycobacter from some of the samples, and 3 is what the animal gets if they've cultured Mycobacter from multiple samples, and or it's got sort of seeping abscesses and things like that that are kind of symptomatic of advanced states of disease. So we can think of this as a kind of progressive score about severity or progression of TB in the badger from 0 to 3. What we know is that there's an awful lot of variation. So there's variation from year to year, there's variation from group to group, there's variation between males and females. It's a bit hard at the moment because this variation could ultimately come about in a lot of different ways, right? So there's probably variation in behavior influencing susceptibility. So, some animals we now know move around much more than others, so they might be encountering more infected individuals, for example. Then on top of that, you've got a layer variation in immune function, which is almost certain to be there. We have some generic immune markers, but I'm not going to talk about those today. And then we might have variation in, for example, tolerance, if you know what that is, but basically how the disease progresses or what the symptom severity is given an infection has become established. So there's different processes that lead into this. We're not currently able to disentangle them, but what we can say is there's a lot of variation. And one of the key things that is emerging is differences among the social groups in what's going on. So the first kind of result I want to show you, this is work led by a PhD student who actually works for the government agency that runs this project, but she's also a part-time student with us at Exeter. And I was only sort of peripherally involved with this, but I'm gonna vicariously claim a bit of her glory by presenting the results. So this is on the y-axis, you've got probability of infection in cubs. So cubs is defined as anything up to a year old. And so obviously whether or not we think a cub it gets infected within its first year. And the first plot there is just showing you that essentially there's no obvious relationship with group size per se. The second plot is showing you that although there's no obvious relationship with group size, the risk of infection goes up as the number of known positive testing adults in the group increases. So this isn't a surprise, right? If you're in a social group with more infected individuals, then you're probably going to have a higher risk. And then the next plot is showing what happens with the number of test negative individuals. So again, it's not a surprise. You have a dilution effect. If you've got lots of negative, uninfected individuals in your social group, then that dilutes your risk. So these two things are just opposing to basically mean that there isn't a big effect of overall group size. So I think that probably is roughly what we'd expect, and there's maybe nothing too exciting about that. But what was exciting that Claire dug out is she had access from some old work that was done right in the early noughties, some microsat genotyping. She had access to microsat genotypes for quite a lot of the animals in her study. And she used some simple relatedness estimators and kind of came up with a cutoff of relatedness of 0.25. So from 0.25 and above, she said, okay, those are related individuals. And below that, let's say that they're unrelated. Now, these kind of relatedness estimators are notoriously messy, so that's probably a relatively sensible thing to do. What she found then is that although clearly the number of infected individuals seems to have a positive effect, so these are effect sizes on the x-axis, obviously 0 means no effect, and the error bars are 95% confidence intervals there. So everything that not spanning 0 is pretty convincing, I guess, at alpha of 5%. And she's broken basically the numbers of group mates into, or the numbers of group mates that test positive at those different stages. So the blood test positive or the excreta, which is the latter stages where you're culturing bacteria, into males and females, related and unrelated. And I guess the things I want you to notice are the bits I've circled in blue and green there. Although the presence of excretors, so that's the ones that we've managed to culture TB from in the group, clearly has an effect of increasing the risk of the cub being infected. That risk appears to be greater if those excretors are actually relatives of yours. So there's something interesting going on here, and this is just kind of showing the same thing, just picking out the males. The argument here is that on the left, where you're looking at the number of excreta male relatives in the group, versus on the right, where it's the number of excreta males that are non-relatives. The slope of that line is steeper, right? So it basically, highly infected excreting individuals pose more of a threat to you, perhaps, if they're closely related. So this is kind of where we got involved, my group, because the guys running the Badger project were trying to think about, well, okay, this wasn't actually what Claire was really looking for at the time. She was thinking about inbreeding, but she found this pattern, and we wanted to think about what might drive it. Now, there's two, I think, very obvious possibilities. The first is that you have kin-biased interactions. So it could just be that hanging out with infected badgers causes risk, and you tend to hang out more with your relatives. Certainly that's possible, and there's some work going on with fitting proximity collars onto some badgers and things to see if that's likely to be true. But the other thing, of course, is that this is totally, could be that this is not a causal relationship at all. What's happening is that if you have if you are more likely to be infected when your relatives are infected, this could be because your risk of being infected is in some way down to your genes, which you're sharing with your relatives. So in other words, your infection risk or progression risk of the disease is heritable. And so the question we were asked is, can we separate genes and environments with quantitative genetics? And of course, my instant answer was, I don't know. The reason I don't know is because what we did know already about the badgers is, although these social groups are not just families, so these are not meerkats, right? There's not like one individual reproducing and everybody's a close relative. But we do know that there's more mating within groups than between. So what we know is that there's more relatedness within groups than between. And that means we have this issue in quantitative genetics, which we call a common environment effect, which is where where relatives share both genes and environments. And that can make it quite hard to disentangle these things without doing experiments. We're working in the field, so we can't do the experiments that we would do in the lab. So the first thing we had to do, and I'm using the royal we here, and I'm going to do something awful, which is boil a previous student, PhD student, Paula's work down into like 2 slides as if it was nothing. And it was absolutely monstrously epic effort. But the first thing we had to do was try and turn this fairly rough and dodgy microsatellite data into some kind of pedigree structure. That was done using a Bayesian analysis that actually allowed us both to use the spatial information, because we had this strong a priori expectation that there are more matings going on within groups than between groups. So we're able to use the spatial information and actually jointly estimate the parentage of individual cubs on a cohort by cohort basis, and these spatial parameters, in particular something called paternity distance, which is just the distance between the main set of where the cub was born and where we think the father was resident. Okay, so you might infer from this that this means males move around to mate. They do, but so do females as well. It's just that the females produce the cub in their own social group, and so we can't infer whether it's the male or the female that moves, but we can think of this as movement of male genes, if you like. Not everything, not all the cohorts in the whole study had microsat data available, so Paula had to go freezer diving back to the 80s, getting tissue where she could, and in the end she managed to genotype individuals from 29 cohorts at 22 microsatellite loci, or rather compile data and supplement it and do de novo analysis as well. We run this through this big sort of modeling process, and we managed to get parents assigned to 34% of the genotype cubs. So that's not an awful lot. In fact, by wild pedigree standards, it's pretty poor. To put that in perspective, there's about 3,000 animals in the database with some information. About 1,500 of those, or maybe a bit more, we managed to get genotype data on. And of those, we managed to assign paternity or maternity to about 600. In most of those cases that we were able to make assignations, we were able to get both the mum and the dad. So that's quite poor, and there's reasons for that. The biggest one we think actually is incomplete sampling. So this is, you can never, on a probabilistic way, you can never assign parents using genetic data if you think there are a large possible set of candidates out there that you never got sampled, right? It's that simple. And so ultimately that poses a big limit. But there are some other particular challenges with badgers. The cubs don't come out from underground till they're about 12 weeks old, I think. So you actually never observe them suckling with the mum, for instance. So we're having to recover both mum and dad here, whereas in most mammalian systems, you know mum and you're just trying to assign dad, which is a whole lot easier. But against that bad news, there was also some good news, and that was that across all cohorts, the mean paternity distance was 354 meters. Now, that won't mean anything to you, but it does mean something to me, because in fact, the average distance between, from one set used by a social group to its nearest neighbor is about 350 meters. So what this implies is that there's actually a reasonable amount of movement of genes between sets. We actually found, I think it was 37% of cubs that had assigned dads had dads that were assigned from somewhere other than their own social group. Right. So this is really what gives us the power to the quantitative genetics. When we do it, it looks like this. So if you just focus for a minute on the plot on the left, so we fit an animal model, and all we're doing now is we're using that pedigree information, and we're using that 0, 1, 2, 3 score to ask how much of the variation in disease state on that progressive score is down to different things. If you focus on the blue lines, I probably should have taken the red ones off. The blue lines is what we think is our best model. And what we can see is there's a little bit of variation down to genetics, so about 0.05 on that y-axis scale, which is actually in standard deviation units. So that's effectively a heritability. And then we also looked at how much is attributable to the social group, but assuming that social group effect is constant in time. So that's really actually a spatial effect. I probably should have relabeled it, but it's really about where the set is, where the animals are living. And it's not too much that's captured there. There's a bit of year-to-year variation, which is shown on the far right. And then in between, we've got a social group by year effect. And that was our way of trying to capture the combination of space and time that actually defines the set of individuals you're living with. So this is what we think is really going on in terms of the social context, the social environment that the individuals are experiencing. And you can see that's about 10%. On top of that, for cubs, and this is the blue lines I'm looking at now, the identity of the mom also explains quite a large chunk, nearly 15% of the variation. The red lines, just for information, are what happened when we estimated all of those parameters, assuming that maternal effects were present at badgers on all ages, and it turned out to be a much better fit for the model if we only allowed those maternal effects to exist on the cubs, basically, which is what the blue line shows. So the table on the left is just kind of showing that nominally, at least, all of those variance components are significant, and it's providing estimates of the heritability. So I don't think you can see my mouse, but it doesn't matter. So if you look on the adult column, for example, VA is represented as 0.052. That's the intraclass correlation, which is another way of saying the heritability. So 5% of the variation down to genes. There's a few weirdnesses about this data structure, and I'm not going to go into them, but suffices to say that we fit models that assume nice things like Gaussian residuals and normal traits that are unbounded, and we know we've really rocked those assumptions here. So we've done a few other things, and we're still working on the models a little bit. One of these is just a Bayesian analysis, which is where we treated the trait as a binary thing. So do you get the disease over your lifetime or not? And what we find then is about 7% heritability on the observed scale, treating it that way. And again, we find evidence of these social environment effects in the form of group or group by birth year in this case, and that's shown the estimate of maternal effects across the whole lifespan, and that's very small, but that's exactly what we expect, given that we think they're only present on the cubs. Okay, so that's the kind of thing that we're working on at the moment, and we're obviously trying to progress that and look at the relationship between, for example, these TB scores and general markers of immune function that we have, and also thinking about the genetic basis of any relationships between TB status and weight loss, which might be a way of looking at the genetics of tolerance. But the bottom line, I mean, a really simple take-home message here is that genes matter, but they don't actually matter that much. Social environments seem to matter much more. And certainly for the cubs, who your mum is seems to be having a big effect. Now, we don't know why. It could be that mums, I told you that infection can last for years and years and years, and individuals can keep reproducing. So it could be that some mums are really bad at posing an infection risk to cubs simply because they're infected, or it might be, I don't know, that they produce poor quality milk. That puts the animals into poor condition, which means they're more at risk from infection from somewhere else. But that's kind of where we've got to. So genes matter, but social environments matter more. Okay, that's kind of a simple and trite observation that we've made there. But it kind of feeds into something bigger, which is that what we've done in that analysis is that we've assumed that genes can be separated from environments, right? We've assumed that the social environment defined by the set of interacting conspecifics can be treated as an environmental effect. So for evolutionary purposes, just a source of noise, right? That's not necessarily true. And so what I want to do now is switch to thinking about whether we might have to worry about genes in the environment. We haven't done that with the badgers, so I'm going to rattle through some ideas and just illustrate with examples where we can say something. Now, I showed you the breeders equation earlier. I didn't tell you our shocking little secret, which is that it doesn't work. It doesn't work at all, really. Actually, that's not fair. It works really well in captive populations in constant environments. It doesn't work well as a tool for predicting phenotypic change in wild populations. Now, that actually shouldn't surprise us because aside from anything else, it's only intended to predict selection response, and most phenotypic changes in wild populations might well be driven by environmental change, right? But more than it not working, what we find systematically, of course, there's exceptions, and the exceptions where people jump up and down and shout about rapid evolution over ecological timescales tend to they end up in Nature and Science. But the times where it doesn't work, which is 99% of them that don't end up in Nature and Science, what we actually find is that given how much heritable variation is out there, or we think is out there, given how much directional selection we think is acting, we actually see rather less phenotypic change or evolution than we might predict given this simple model. So it's more a problem of stasis or constraints on evolution, I think, than trying to, focus on the super sexy, exciting, rapid evolution cases. Maybe that's just because I'm a glass half empty kind of pessimist guy, and I should focus more on the exciting things. But there we go. So this is something that Luska Crook wrote. I say she wrote it because I'm distancing myself from it, but the quote actually you'll see is from myself, John Slayton, and Luska. But Luska wrote this particular line. It's probably fair to conclude that we are currently less confident about predicting or understanding temporal trends in the phenotype than we might have thought we were 10 years ago. She wrote this 12 years ago now. The bit I cut off from that is that the end of the sentence was, clearly this is disappointing. And I thought about it and I thought, well, I cut that off because if I'm completely honest, if we'd nailed it 10 years ago, I wouldn't have a career. So actually what's happened in the last 10 years is we moved from thinking we could predict this evolutionary change using simple models to trying to understand why we can't. And of course, the interesting biology is not in showing that a simple model doesn't do a very good job of explaining the world. It's actually in trying to work out when, what, and why things are missing from that simple model. So what's the important biology that we're missing out? And there's lots of things that we're missing, for sure. That's a univariate model, and actually, we think, for instance, that selection doesn't act on single traits in isolation, it acts on multivariate phenotypes. I think that's probably the biggest one. But for the rest of the talk, I'm going to focus on the possibility that I've already alluded to, that one of the really big things we're missing from that simple model is social interactions. So we know that individual phenotypes are often socially plastic. So what I mean by that is that individuals adjust their phenotype plastically according to traits or traits being expressed by some kind of social partner, right? That might be a mate, that might be a rival, that could be a parent, or that could be an offspring. This is an example just from one of our lab studies. This is a green swordtail, Xiphophorus helleri, and all I'm showing you here is the latency to attack an opponent by a focal individual on the y-axis, and you can see that that depends on how big the opponent is, right? So in simple terms, if you give this focal individual a really big opponent, then suddenly it's not such a big man after all, and it's going to sit quietly in the corner. Whereas if you give it a small opponent, it's much more likely to rapidly attack and chase that opponent. So what we can see here is that the behavioral trait, in this case latency to attack the opponent, that we've decided is of interesting to us, is clearly influenced by the opponent phenotype. Now, the reason I showed you this example is that of all the traits that we could ever think about, the only one I could pretty much guarantee is going to be heritable or substantially heritable in almost any study system is size, right? So we can see here that behavioral traits expressed by the focal individual are causally dependent on body size, which we know is heritable, and so causally dependent almost certainly on genes expressed by the rival or the opponent that we introduce into the tank. So this is just an example. We didn't do the genetics here, but it's an example of a phenomenon that's been called indirect genetic effects. And this schematic is kind of nicked from an old paper of Jason Wolf's, actually, but it's replicated in similar forms in many other places. And it's just showing you then that we have a causal pathway between the genotype, so G of one individual, J, which I'll call a social partner, and the phenotype, Z, of another individual, I, right? So the point being here that we've been thinking all the time about how the phenotype of an individual depends on its genes and its environment, but actually its environment includes contributions from the genes of its social partners. So where are these going to occur? Well, they could occur almost anywhere, right? So this is where the ecologist in you starts kicking in, and you're like, well, where wouldn't they occur if you really start to think about it? But the most obvious places will be behavioral interactions with other individuals. So the key places, we could start to look at, we could think about things like parental care, we could think about competition, which in this case might be among relatives, might be among unrelated individuals, and there's some nuance to that. But we can also think about cooperation. We could think about aggressive contests, so fights and things like that. But we can also think about mate choice and reproductive traits, right? It's bound to exist, right? We know that individuals of one sex respond to traits expressed by the other sex, right? We know that already. So it seems almost inevitable that these things are going to be here. There was a kind of review by Nathan Bailey and Lucas and Alan Moore, who some of you may know, a couple of years ago. And they made the case that behavior is special. So if you're interested in behavioral traits, you really need to think about these indirect genetic effects, I think. And they made the case behavior is special and bound to be influenced by indirect genetic effects, because of course behaviors are what mediate these social interactions that we know are so important. Now, although that seems inescapable, I actually think Nathan's completely wrong here, because yes, behaviors are bound to be influenced by indirect genetic effects, but behaviors also mediate what happens downstream, right? So by begging, you elicit more food to be provided, say from your mum or your dad, and you get more food and you grow faster. So the pathway here might be your behavior influences someone else that then gives you more food. So there's an indirect effect from the mum, say, to the offspring, but it's going to impact growth, right? The point is not to get more food just, as some kind of tick a box. It's because food is a limited resource, and food gets translated into growth, into reproduction, into survival. Okay, so I think Nathan and Lucas and Alan were wrong to suggest that IgEs or the behavior is special because of IgEs. I think that IgEs are the easiest place, behavioral traits are the easiest place to find the IgEs, but that their influence is going to extend far beyond that to all sorts of traits that we don't normally think of as social traits. So what are the consequences of it? Well, let's break it down a little bit. The first thing to say is that in the presence of indirect genetic effects, if we were to assign a phenotype or a trait to a focal individual, in this case, let's think about the growth rate of this offspring, what we need to recognize is that genetic control of the focal, in this case, offspring trait, is shared, right? So this is no longer a genetic property of one individual. We might observe it on one individual, but it's a shared genetic control. That also, another way of thinking about it, and it's kind of the other side of the same coin, is that environmental effects contain genes or are shaped by genes. And so if you've got genetic variation in environments, social environments, then social environments can evolve, right? And that means we're going to actually set up co-evolutionary dynamics between social environments, however we want to kind of measure them, and focal traits, in this case, growth. And those co-evolutionary dynamics, we know a bit about co-evolution already. We know that co-evolutionary dynamics can act so that they can actually accelerate. You can have sort of runaway processes that lead to rapid evolution of the focal trait, in this case, offspring growth, but they can also act to dampen, constrain, or oppose the change that you might expect if you don't think about that co-evolutionary process. And this really is the nub of why IGEs are interesting, because they will fundamentally change our evolutionary dynamics, right? We have an extra source of genetic variation in the system that we haven't been thinking about, but that extra source of genetic variation is not always going to accelerate selection responses. It might do that sometimes, but sometimes it might actually dampen them down and constrain them. So I think I might be right in saying that Ben Dancer gave you a talk recently. Matthew, I'm still looking at you. Is that true?
Next week.
Right, you don't have to answer. You can just put your thumb up or down.
Next week.
Next week. Okay. So, okay, great. So I decided not to talk very much. I'm gonna mention some squirrels a couple of times, but I'm not going to tell you any background about them, because I thought either that would bore you because you'd heard it, or spoil Ben's talk. So the first person or first people who really showed IGEs in a wild long-term study were actually the Kluane Squirrel Project, and this was work actually led by Andrew McAdam, who's now at Boulder, I think, just moved from Guelph. And they showed that juvenile mass in red squirrels is weakly heritable, so heritability of about 10%. So that's just thinking about the influence of an individual's own genotype on its own trait. But they also identified that there were strong maternal effects in there, and they showed why they thought those maternal effects themselves had a genetic basis, and argued, in effect, that the maternal effects will increase the evolutionary potential threefold. So this is a system where they made the case that the indirect genetic effects, in this case in the form of maternal effects, could actually lead us to underpredict the response to selection. There's some old theory. This is from the work by Willem in the 60s and then leading on to the 70s, that kind of gives us an adapted breeders equation. And the things I guess I want you to notice is that we've replaced heritability with this h squared with a little t. T stands for total, and total then is what Willem referred to as the total heritability. And you can see it's something in brackets divided by the phenotypic variance. So that's the sigma squared p. So again, it's just a proportion of the phenotypic variance. But the things going into that, the first term is the normal additive genetic variance. So that's the variation in focal trait caused by focal genotype. The third term is half of the maternal genetic variance. So that's how much variance is caused by the mum's genotype over and above effects of genes that are passed on directly to the offspring. Okay, so this is genes being expressed by mum at the time. And then in the middle there, you've got 1.5 times this ugly looking thing, which is actually the covariance between the direct and then the indirect effects. Now, covariances, you don't need to know really much about them, except that they can be positive and negative. If they're positive, you can start making this number really big, and certainly it can be a lot bigger than the direct heritability. But if they're negative, it could get really small, right? And it could actually be so small that in a weird way, it's perfectly possible with maternal effects to select a trait to move in one direction and for it to actually, over a single generation, go the wrong way. That's kind of shown in this schematic. I won't go through it too much, but this is from an old paper by Jason Wolfe, and it's just predictions from a model, so it's not data. And it's saying what would happen if you selected on an offspring trait, let's say it's growth, it doesn't really matter, with or without maternal effects, and what does the influence of that covariance, or what could it do? So you can see actually the pattern's not too dissimilar across these three plots. What I want you to actually do is notice the scaling on the y-axis. So in the first plot, there's no maternal effects. In the second plot, Jason added some numbers that were fairly plausible for a magnitude of maternal genetic variance, but with a positive covariance or correlation between the direct and indirect genetic effects. And you can see that over 8 generations of selection, you get, in this case, a 4-fold increase in the amount of change, evolutionary change that you managed to achieve. And then in the third case, you made a negative. So again, you got maternal genetic effects, but you've made the correlation negative. And what this does is dampens down that net response. So you can see that by generation 8, it's only gone up to about 0.5. And the other thing you can see is these weird dynamics here, right? Where you start seeing the phenotype bouncing up and down. And you'll notice that over the first generation of selection, the offspring trait actually went down despite being selected up. So they can do weird and unpredictable things. We looked at maternal genetic effects in the Soay sheep as well. This is just because the last bit was a little bit heavy and I thought we might need some pretty pictures by now. So there's some pretty pictures of some sheep, and this is just to establish my field credentials. I have actually done field work on a few of these projects. The one I've done most field work on is the Soay sheep, where I spent quite a bit of time out on St Kilda. That's me with Josephine Pemberton there a number of years ago. Yeah, lifting sheep, which is what I was good at, it turned out. I wasn't very good at more complicated things, but lifting sheep I could manage. So we were able to explore maternal effects and partition out, using animal models, partition out the proportion or the portion of the maternal effects that we thought were genetic in that case. And actually the answer is not so dissimilar to the squirrel example. About 8% of the variation in lamb weight, so this is weight in August after they're born in the spring that year, is attributable to their own genes, but about 12% is attributable to mum's genes, which isn't surprising, right? Because mum's probably expressing lots of genes that influence both uterine provisioning, but also in this case, the period of which we're looking would have captured postnatal care. So you think about genes influencing milk quality or quantity and all sorts of things. But there is a limitation here, which is that we don't know what's driving this, right? So as quantitative geneticists, it's all very well for us to slice up the variants and say, yeah, that's coming from there, that's coming from there. But as biologists and ecologists and behavioral ecologists, we actually want to know what it is. And this is super challenging because with parental effects like this, we suspect that behavioral interactions are key, but getting high volume data on weights is a lot easier than getting high volume data on behavioral interactions between, say, mothers and lambs. This is a plot from a PhD student, Charlotte Regan, who actually used, so the Soay system is quite sophisticated now in terms of genetic resources. So Charlotte was able to use high density SNP data to actually come up with genomic predictions of genetic merit for individual mums. Then what she did is went out in the spring, saw which mums were still alive that year. Then she saw where they were living and she worked out whether she could do a targeted sort of focal watch type behavioral study on a set of females that were still alive that had either high or low predicted genetic merit. And what she did was to compare a suite of traits broadly around suckling time and suckling interactions and things like that. And there's certainly a hint here that for the group on the right, which is the group of mum-lamb interactions where the mums were predicted to be genetically poor at creating heavy lambs, you get a lower proportion of successful suckling events. So what's going on here is the lamb is coming in and attempting to suckle from the mum, and either the mum allows the suckling to happen or she kind of steps away and the offspring has to go hungry. So we're maybe beginning to start to unpick how these effects are coming about. But data is limiting. And I think actually as field biologists, this should be super reassuring because this is a massively genomically well-resourced project. But in this post-genomic era, it's the phenotype that's key here, right? So we haven't been able to really nail this because we can't get high volume behavioral data. But then there's other things we might want to go after, like physiology, like milk traits, like all kinds of things. But again, the sampling to do in the field and to get those samples is really hard, and that's kind of limiting. But we can start to explore mechanism, I think, in some interesting ways. Okay, so I kind of mentioned you've got that negative covariance idea, and that could reduce the amount of total heritability in the maternal genetic effect scenario. So let's think about whether this could happen more generally. So forget about maternal effects or parental effects. Let's just think about individuals interacting. And in fact, let's think about unrelated individuals interacting. Relatedness is important. Relatedness matters for all forms of social evolution. But in quantitative genetics, we tend to put it normally into, we tend to work out how much genetic variance is there from direct and indirect genetic effects. Then we work out the selection, and we moderate the response according to the relatedness structure. So I don't want you to think that we've completely abandoned thinking about relatedness. It's just in a different part of the model. So this is a generalized version of what I've already shown you with some slightly different notation. So TBV stands for total breeding value. Think of this as just the total genetic variance, and it's now equal to the sum of the direct, that's the D, additive genetic variance, that's the thing on the left. On the far right, you've got n-1, so n is the group size, all squared, times this notation means the indirect genetic variance, not particularly, don't worry about remembering the notation because I'm going to throw it all away in a minute and show you something simpler. But this is the original model. And then in between, you've got that covariance as well. And you can see that there's an n-1 in there. So you're starting to multiply things up by effectively group size. Remembering that covariance could be positive, it could be negative. All right, imagine you have negative covariance and big group sizes. You can quickly swamp the indirect genetic effects, very quickly become a much more important driver of evolutionary dynamics than that direct heritability that we're all used to thinking about. So let's just strip it down. Let's imagine we're just working with two individuals. And I'm going to show you some examples where we've been working in a kind of contest scenario. So I'm going to talk about a focal, which is the individual we watched, and an opponent, which is the individual it was fighting or playing against, right? So for a dyadic case, and I've just switched sigmas. So if we're being all proper about things, variances and covariances, we use sigmas. If we've been a bit lazy, we can use V's and COV for covariance. So this is the same thing for the case of a group size of 2, a dyadic interaction. Total genetic variance is equal to the additive, direct additive variance among focals, plus the opponent variance, plus twice the covariance. Again, that covariance can be positive or negative. And if it's negative, we could end up with a much lower value here than we were expecting. Okay. I thought it would be fun to go after these and see if we could do this in wild populations. And the idea really is that theory predicts under competition, IgEs will be negatively correlated with direct genetic effects. In other words, competition, although it's an ecological process, will tend to come up with negative numbers in that equation that reduce the amount of genetic variance available for selection to act on. And so actually this might be a reason why we see a lot less evolution than the simple models predict. This could be true in any kind of competition. And classically, we might think of a continuum from scramble competition where individuals can't exclude competitors, everyone's, say, eating as fast as they can. And we tend to sort of collapse this down to density dependence. Everybody's equally good, everybody's equally bad. We can just think of it as a numbers game. But if you think about animal contests and all that classic theory out there, it's rather different, right? So in a contest type of competition, the winner tends to get all the resource and the loser gets nothing. So it's a very asymmetric division of outcome resource. We also know from all the classic work that there's loads of assessment strategies and things like, you know, the classic stuff on resource holding potential. It does matter. What you do depends on how big your opponent's horns are or their body sizes or things like that. And we know, of course, that those morphological traits are highly heritable. So the reason we went after a dyadic contest competition is that it's tractable. We're dealing with group sizes of 2. It's likely to cause big effects because winners get resource and so fitness at the expense of losers. And we already had the a priori expectation that the traits involved in mediating those would include these heritable kind of aspects of morphology. So in other words, we did what all good scientists should do at an early stage in their career, and that's go after the very lowest hanging fruit you could see. And so that's what we did. And I'll kind of just show you a few examples. This pathway is just to illustrate the wider thinking here. So imagine you've got some contest behaviors. So you might be looking at aggression, you might be looking at duration of contest or whatever. But ultimately, those behaviors feed into whether or not you win the contest. I'm going to use the term dominance, which means different things to different people. So I'm going to clarify it now. If you're used to thinking about stable dominance ranks in complex societies, that's not what I mean. What I mean by dominance here is consistent, repeatable ability of an individual to acquire, win, or hold on to resources. So in other words, its ability to win contests. In a complex society, then dominance rank might be the best way to measure it. In an animal contest experiment, you could think of it as equivalent to resource holding potential. Actually, in a scramble competition scenario, it might be something as simple as straight line running speed, right? Who gets there first? But so it's whatever you need to do to win the competition. And individuals that consistently win the competition are socially dominant in the term that I'm using. Being dominant means you acquire more resource. Acquiring more resource means you can plug more into all of your resource-dependent traits. And critically, that is everything we think is really under strong selection, right? It's growth and it's all your life history traits. That's the pointy end of where phenotype meets fitness. Okay, we have focal genotypes, so direct genetic effects, which create variation all the way down this pathway. And that's the variation we're used to thinking of as the additive genetic variance that we standardize as the heritability. But because it's a dyadic contest, we can have an influence of the opponent genotype that generates variance all the way down here as well. So what can we do? Easiest thing to do, we kind of came up with this idea. Actually, I was visiting Denis Rial, who's another long-term study guy up in Montreal, and he had some data on deer mice, so peromyscus, that he had caught and brought into the lab and done a bunch of dyadic contests. And we thought we would test this idea in his contest data, where the critical thing was, for every contest, the identity of the focal and the identity of the opponent, and they're both contained within a pedigree, right? So they're related to each other. So they may or may not be related to each other. And every individual is used in some contests as the focal and some contests as the opponent, right? So the designation is somewhat arbitrary across contests, but within a contest, you know who's who. So just to give you one example, rearing rate. So this is the rate at which the mice sit basically on their haunches and get up, trying to kind of look over or assume a dominant position over the other individual. The direct estimate of the heritability is only 10%. Under the indirect genetic effect model, where we fit both genotypes and model it as a shared trait, and then use that equation I just showed you, we actually come up with an equivalent proportion of phenotypic variance that would be responsive to selection of 60%, right? So if we could make a straightforward switch of into our breeder's equation, it's a bit more complicated than that, but let's just for a minute imagine we could, we would increase the expected rate of evolution by sixfold. And that's because of a very strong positive correlation between the direct and indirect genetic effects. Oops, I think I've gone the wrong way. So that's cool, that's exciting, but that's the opposite of what I told you. I told you that competition was actually going to tend to cause constraints on evolution. So let me show you exactly why that is inescapable. This is data from the red deer study, about 10,000 dyadic contests featuring 1,200 deer. These are not the massive iconic stags fighting each other. These are feeding interactions where one individual displaces another. Okay, so you might be thinking it's very dramatic. It's not quite as dramatic as that. What we're able to do, though, is if you think of that as a contest and you go, okay, who wins most of the time? And we can show, for example, that winning is repeatable. So there are sort of hierarchies of feeding, if you like, which we've characterized as dominance. And it turns out that if you're more dominant, you win more contests, you have higher fitness, lifetime breeding success. So if we make that kind of cardinal assumption that correlation can be used safely as causation here, it appears as though dominant individuals do better, which is exactly another way of saying that the trait is under positive directional selection. If we run that through an animal model and work out what the heritability of contest winning is, it comes out at 10%. So this is fantastic. We've now shown that winning a contest is heritable and under selection. If we plug that into our breeder's equation, we're going to predict that mean contest winning will evolve, right? And hopefully by now, some of you have spotted the obvious absurdity of this position, which is that if you have a dyadic contest with one winner and one loser, the mean phenotype, as we've defined it, will always be 0.5. So how do we square that circle? We square that circle by recognizing that this contest outcome cannot be a trait of the focal individual. It has to be a shared trait. So although genes are important in determining the contest outcome, we can't just assign this to whoever we've decided will be the focal individual. So the model we use, the sort of extended animal model, looks like this. It doesn't really matter, but we have an outcome. This is binary data, so there's an extra level of complexity that we don't need to worry about. But basically, your outcome 0 or 1 is predicted by some fixed effects. So we have some effects of age and sex in there that we're just trying to take out. And then what we have is the genetic value, the additive genetic value of the focal individual I, plus what we call its permanent environment effect, which is the non-genetic repeatable thing. So any part of the consistent tendency of that individual to win that isn't down to genetics. But then we also have to have the same for the opponent, okay? Because what we see is necessarily an outcome of the two individuals. And that then captures the direct and the indirect genetic effects. So if we fit that model, right, we can extend our animal model, fit that, and we say, well, does it fit better than the normal one where we assume all the genetic variance is just from the focals? And the answer is yes, it does massively, right? So statistically speaking, there's no doubt it's a better model. So we can then plug it into our equation to see, well, how much genetic variance is there available for selection to act on then, right? And this is the equation we had for the dyadic situation. And the answer comes out at 0. It comes out at 0 to, actually, I was really proud of this because to 3 decimal places, it was 0. So this is super cool because we can recover this empirically, right? And we can do this statistical analysis. We can fit the right models. We can show the models are better, and we can come up with an answer that allows us to reconcile quantitative genetics with common sense. And that's actually not something that gets done that often. So I'm quite proud of that. Now it has to be true. So it has to be true because the mean contest winning has to be 0.5. But that's the simple answer. Let's try and see if we can get our head around the complicated way of kind of rationalizing that. And it works like this. Any genotype that predisposes to winning when it's carried by the focal individual has to predispose to losing when it's encountered by a focal individual being carried by an opponent, right? And because the designation in this case of whether you're a focal or an opponent is arbitrary, it actually also has to be true that the variance in the focal genotype effects has to equal the variance in the opponent genotype effects. And because the designation is the same, the genotypes having the same effect, it's just the sign of it that's going to flip. So the correlation between those also has to be perfect, has to be -1, which means that the covariance has to be the same magnitude as the variance, just with a minus sign in front of it. So in other words, it has to be true that these terms will add up to 0. But it doesn't have to be true, of course, statistically that you can recover that truth, right? I mean, there's statistics and there's data and there's the real world. But this is nice because it basically allows us to find a way to reconcile quantitative genetics, as I said, with common sense. And one of the things that remember I said about quantitative genetics is it's applicable to any trait, right? Any trait in any sexual diploid. And here we decided that contest winning is a trait. That was our decision. But quantitative genetics is applicable and it does work, provided you recognize when the genotype or when the phenotype is really inevitably shared. So that's a really trivial result. I also told you that quantitative geneticists like to come up with complicated explanations for simple things, and that's a classic case in point. So here we go. There has to be an absolute constraint on the evolution of dominance if we define it as contest winning. So let's just take a step back and think more kind of generally about what's going to happen with these IGEs. What's actually going to happen is that IGEs could either facilitate or constrain the evolution of traits like the behaviors, like the rearing rate in mice, that are involved in determining the outcome of that competition. So some traits might run away under selection, some might be constrained. But the contest outcome and the dominance has to be constrained. That's inevitable. And what that means is that to the extent that resource acquisition is determined by competitive interactions, there will be a load of variation in resource acquisition that is caused by the genes individuals are carrying for contest winning. All right, so if you have genes for winning contests, you have genes for acquiring lots of resource. You have genes for plugging lots of resource into your competing life history traits. But it also means that actually all of these traits downstream on the bottom part of the panel here are going to be subject to constraints from indirect genetic effects. So only the part of the genetic variance down there that is independent of this pathway can actually contribute to selection responses. So what that could mean, if competition is super important, is that while we've been estimating all the genetic variants down there, we actually need to estimate only the part of the genetic variants that is independent of this pathway. Or equivalently, we have to fit the indirect genetic effects downstream, and what we should find is that the total genetic variance available for selection is rather less than our simple estimates of their heritability. Does it work? Maybe. So again, another lab study, we know this is another Ziphophorus from the Sawtail group, we know that individuals that grow fast do so at the expense of their tank mates and repress their growth, and so those social phenotypic effects are true. If we work out how much of the genetic variance is independent of how much genetic variance in growth is independent of that pathway, we can reduce our heritability from 22% to 9%. So effectively, we're predicting a reduction in selection response of more than half. Is it important in wild populations? Really, generally, truly, we don't know. Certainly, competition's ubiquitous. Certainly, we find this widespread problem where response to selection is less than the simple models predict. And we also find positive genetic correlations among life history traits. So standard theory, if you read Stearns or things like that, will predict that there's negative genetic correlations among life history traits. Actually, if there's genotypes out there for winning contests and getting lots of resource, those genotypes will allow you to put more resource into all of your competing traits. And so we actually expect it to be positive ones. But what we're really missing is an empirical demonstration of indirect genetic effects on life history traits. I think the closest we've come, again, I'm not going to talk much about the squirrels, but the closest we've come perhaps is a study by Dave Fisher and others on the Klawani project, where what they try to do is capture the real complexity of competition in the field in the sense that individuals are not just going through a series of dyadic interactions, they're embedded in some kind of network. They were able to show that there were indirect phenotypic effects, in this case on reproductive timing, of living near strong competitors. Squirrels are territorial, I probably should have said. And they were able to show that breeding time has heritable variation, but when they tried to fit the indirect genetic, when we tried to fit the indirect genetic effect models, we could fit them, but the model just blew up in terms of the uncertainty. So we could say yes, we've estimated them, but we certainly can't prove they're really there. And I think this is kind of the challenge going forward, is how do we take these ideas and translate them to real world situations? Now, I suspect I've talked for a long time, haven't I, Matthew?
Pretty long, about 80 minutes.
Oh, crikey. Okay, so I've totally been waffling. Okay, so I'm not going to talk any more about cooperation, and I'll just wrap up. I guess if people are interested in cooperation, they can always ask me what I was going to say later. So I'll just stop there and I'll summarize what I've been saying. So quantitative genetics provides us with the general framework for studying trait evolution. Long-term individual-based studies provide the opportunity to apply this in the wild, which has been fantastic and really generated a lot of movement over the last couple of decades. We can separate genes and the environment, and this tells us about evolutionary potential. I just kind of gave you a univariate example, but of course it gets more complicated than this. But caveats to that are that separating genes and environments in the wild are not trivial, precisely because we typically have to abandon all our normal experimental checks and balances, and that poses us problems. And these simple predictive models often fail very, very badly in the wild as well. So one reason why they might fail is indirect genetic effects that arise from interactions in the social environment. IGEs can both accelerate or constrain selection responses, depending on the circumstances. We do know that they exist, right? We can show them in parental care and we can show them in wild populations in other dyadic contexts. But can we go further than that? Can we think about whether they really do pose a general constraint on life history adaptation in competitive situations where we really try and model the ecology, with all its glorious complexity? And I'll leave you with the intriguing thought that IGEs actually might be as important as relatedness in shaping the evolution of cooperation. And with that, I'll stop. I'll just flag the people I need to thank. The names in bold at the top are all the early career PhDs and postdocs that led the papers I actually was talking about. I thank you for listening, and I apologize for talking for so long. I had no idea.
Well, thanks, Alastair. That was really, I mean, it was long for sure, but it was really, really interesting. And I think all of it was really helpful, at least for me, for increasing my understanding of how this works. I had a question, and maybe we'll just leave it with this one question for today.
Yep.
Which is when I was first introduced to the concept of IGEs, I found them just extremely exciting because, you know, I think it really does capture this disconnect between the classic quantitative genetic literature and how we think things work in nature. And so I'm a bit biased because I'm trained on baboons. So I think about social groups that are large with and not constant, changing over the course of an animal's rather long lifespan. And the result is that the social environment at any given time and the indirect genetic effects that might matter at any given time are extremely variable over an animal's lifetime. And so like in my mind, what I always want to hear about is if you think about the 45 animals in a group, right? How do they affect the focal animal's phenotype? And I think we're probably pretty far from being able to answer that question at this point, right? And so I'm curious what you think, what kind of data we would need in order to really understand the impact of a large number of group mates on an animal's phenotype. What would that data look like? And what are the prospects for being able to kind of get to that point?
So, okay, so what the data would look like, first of all, is it would be a big data set. I mean, that's kind of the first thing to say. Actually, so if you have things like a social network, for example, and we can take these IgE models and we can combine them with social or spatial networks, and we could use those to moderate our expectation of the extent to which individuals experience IgEs. This actually is what Dave did in the squirrel paper. There might be 400 squirrels alive at that point in time, but he decided that actually it's biologically sensible to say that squirrels are only interacting with their 5 nearest neighbors, right? So you can weight the expectation. So I think the way forward is to reduce the size and scale of the problem by, of course, this won't apply to every situation, but potentially I think to find systems where we can say, look, there's 5 to 10 nearest neighbors or most common associates that are likely to have the pronounced effect, and then we've reduced the scale of the modeling problem. So that pragmatically, that's the way forward, I think. But the other thing that has to happen if we're going to accept these studies, and I'm not saying we should or we shouldn't, but we also have to accept that more complicated models come with higher uncertainty. So do you want your best estimate of what would be going on if you believe this model, or do you want a small p-value? Because I don't think these are going to be easily compatible outside of experimental systems, but we know from lab simple experimental systems that IgEs matter. So the question now is, do you want to prove they matter in your system, or do you want to work out what would happen, if you believe the model? And that's going to mean, being a bit more mature about p-values, I think.
Great. Well, thank you very much. I think that was really an insightful talk. And I'm going to go read that squirrel paper right now. So,
okay.
Thanks very much.
Thank you.
For everyone out there, next week, we have Ben Danzer, same time and place, to wrap up the summer version of this series. So thanks very much, Alastair.
Okay. Take care, guys.
Bye-bye.